All right, it's just 11 now. I guess we'll give it maybe a minute or more here and see who else joins in before we get started. Um, but I guess, Heather, are you sharing the presentation or am I sharing the, if you want
I'm
to get
happy
started?
to.
Okay. Probably better if you do anyway. My computer's got a bit of a lag to it.
Can everybody see that? I'm going to go to a full screen view. Okay. And we would just ask that everybody please mute yourselves while Diane is speaking so that we don't have any interference or background noise. And Diane, you can take it away whenever you're ready. Nicole, did you have anything else you wanted to share before we get started?
I guess just really quick here, so everyone is on the same page with how today's going to run. We've got two talks today. First up is Diane Stoecker, followed by Kevin Flynn. They'll both talk for about 15 minutes-ish, and we can have a couple minutes for questions afterwards, but try to keep the questions more just like for any clarifications about things from the specific presentation, because then after that we're going to go into breakout sessions and then discuss this topic in groups for about 30 minutes or so, and then we'll come back together in the end and have a oh sorry my computer might be a little bit choppy. Sorry if the sound's not great. Um, but we'll come back together around noon, and we'll talk about just have a quick report about what each uh, group discussed, and then wrap up from there. So maybe some last group, little short group discussion, wrap up, and finish up around 12:30. So we're looking at about 90 minutes, a full 90 minutes. But we'll let Diane start off here then with talking about uh, definitions of mixotrophy, not including Mitra 2016.
Okay, thank you, Nicole. Could I have the next slide, please? Well, what is mixotrophy? I'm getting it up on mine too. Traditionally defined as carbon acquisition using alternative modes. Um, but maybe looking at just carbon is too narrow. I sort of prefer a very broad definition of mixotrophy, just being autotrophy plus heterotrophy. And that can include not just carbon, but nitrogen, phosphorus, trace elements. In the ocean, iron may be particularly important, and trace nutrients like vitamins and energy. So one thing, it doesn't really have to do with the definition, but I think it's important to remember is that phototrophy is not just carbon fixation. It's also acquisition of chemical energy, um, assimilation of organic nutrients. So in mixotrophs, um, phototrophy can involve the use of light energy to produce chemical energy, photoreductant, with or without carbon fixation, and may not always go together. Um, and it may or may not involve uptake of dissolved nitrogen and phosphorus because these elements in a mixotroph might be provided by ingestion of prey. Have the next slide, please. Well, mixotrophy in, I think this session, we're mostly interested in mixotrophy in planktonic protists. But I think it's important to remember that mixotrophy is actually quite widespread. There are mixotrophic bacteria, archaea, protists, invertebrates, and plants. In mixotrophic bacteria and archaea, the heterotrophic mode is osmotrophy, and the autotrophic mode can be phototrophy or chemoautotrophy. Most bacteriologists, microbial ecologists who work on bacteria don't use the term mixotrophy, but some of them do. Mixotrophy can also be very important in cyanobacteria, including planktonic Synechococcus and Prochlorococcus. And this has attracted more attention recently, particularly in ones that live in low light environments. The other thing to remember is that, well, in Protists and Metazoa, usually we use mixotrophy, and we sort of reserve that term for the combination of phototrophy and phagotrophy. And Kevin will probably go into the reasons for doing that, but very briefly is most protists can take up some organic carbon, and so it's really not useful in discriminating strict autotrophs from mixotrophs among the protists. But even though we sort of, for now, disregard it from our use of mixotrophy for metazoan plankton and protozoan, protistan plankton, is it actually in some groups, we're not sure, but may have some importance. So we shouldn't throw it away. And actually in biotechnology, when they talk about mixotrophic flagellates, they're talking about usually phytoflagellates that can take up dissolved organic material. And this is important in like in investigating biofuels production and you know, like cleaning up wastewater and things like that. So in other fields, mixotrophy is used somewhat differently than most oceanographers use it. Originally, ecologists and aquatic ecologists presented mixotrophy as a spectrum or a gradient from, okay, you were a phytoplankton, you were a complete autotroph, or you were a mixotroph that was someplace along this gradient to complete heterotrophy. And so that, you know, you could sort of present a mixotroph, well, they're 50% 50% autotrophic or 20% autotrophic or something like that. But actually that really doesn't work very well because mixotrophy is qualitatively as well as quantitatively different in different organisms. And mixotrophs really differ among themselves in their biology and their physiological responses to important environmental variables like irradiance, prey availability, and inorganic nutrient availability. And so this led to efforts to group mixotrophs into what we would now call functional types. Have the next slide, please. On my screen, I come up looking at the things I have to look on another computer, so that's why it's a little slow here. Well, the first person to really classify mixotrophic protists by their sort of physiological type is Jones, and this is Harriet Jones, a distinguisher from Roger, who has also worked on mixotrophy and flagellates. But Harriet Jones looked at growth responses of mixotroph to changes in irradiance and prey density. So essentially, she did growth rates with prey and without prey over time at, let's say, different irradiances, and therefore you could get a response curve to these different changes in irradiance and prey density. So hers was also very conceptual because there isn't very much data except for a very few species. And she worked with only flagellates. She worked mostly with freshwater and estuarine flagellates. And importantly, she included additive as well as interactive responses. And this is important because in mixotrophs, I think the early work on gradient, where they thought of them as being on a gradient, well, you have so much photosynthesis and you have so much phagotrophy, let's say, and you just add those two together, the amount of carbon they get from, and you get, what they're getting so for their metabolism. But now we know that for many mixotrophs, the effects are really interactive in that, let's say you're nutrient limited and you eat, you're in a mixotroph. Well, the nitrogen and phosphorus you get from that may allow you to be more photosynthetic, so you can have additive responses. And Harriet Jones, she only included flagellates in her study because essentially that's what there was data on at the time. Can I have the next slide, please? Just to show you her groups. Group A was mixotrophs where growth was dependent on heterotrophy. And so phototrophy supplemented the growth rate, but they couldn't grow phototrophically without eating. Group B is primarily phototrophic, and these would only feed when light was very limiting. And an example of that is many of the Chrysochromulinus species. Group C are primarily phototrophic, and they feed to acquire essential growth substances, such as some phospholipids or iron or vitamins. And then group D was primarily phototrophic, and they had very low ingestion rates, and they seemed to only feed when they were severely light limited. An example of this would be Cryptomonas ovata, a freshwater species. So, and her conceptual three-dimensional pictures of these are really based a little bit on data, but they're really conceptual, how she thought they operated. Have the next slide. Well, a year later, I published a paper on classification of mixotrophic protists, and I was influenced by Jones's work. Her paper was already published when I published this. And I divided them up somewhat differently. I also used a physiological basis for this, but rather than using growth, I used responses of photosynthesis and ingestion to changes in irradiance, nutrient availability, and prey availability. And I also included, besides the flagellates, ciliates, and rhizaria, because all three groups, broad groups of protists, are important in the oceans. Well, type 1 mixotroph, I called the ideal mixotroph with balanced phototrophy and phagotrophy. And it's sort of interesting that this doesn't seem to exist very often in the oceans. The closest species I came to that sort of fit this was Fragilidium subglobosum. It's a dinoflagellate that can eat serratium, another dinoflagellate, and Fragilidium can grow phototrophically. It can grow phagotrophically, and the growth rates are fairly similar, but it grows better mixotrophically. The next group I used was type 2, and I used the term Phagotrophic algae for this. And these are organisms that are primarily phototrophic. Type A feeds when dissolved in inorganic nutrients are limiting. And many of the bloom-forming dinoflagellates, at least in coastal waters, fit this description, and often it's phosphorus limitation or imbalances between nitrogen and phosphorus that induce feeding. Type 2B, feed when trace organic materials are limiting, and the same groups that were found before to induce feeding in these groups are things like uroglena and other chrysophytes. And then type 2C, feed under light limitation, such as chrysochromulina and the Heterocapsa rotundatum data that Nicola's worked with. So then type 3, I defined as photosynthetic protozoa, and you probably wouldn't use this anymore because you really shouldn't use protozoa, you should use protists. And we really know now that the algae and the protozoa are all protists. But I use this for organisms that were primarily phagotrophic. So the first type, 3A, fixed carbon when prey are limiting, but they have their own plastids and can assimilate dissolved inorganic nitrogen and phosphorus. And an example would be Proteochromonas, one of the groups that Harriet Jones also listed in hers. And then the type 3B is protozoa with acquired phototrophy that either have endosymbionts, sequester plastids, or take up a bunch of organelles like mesodynium from their prey. And these, I said, fixed carbon to supplement their carbon nutrition. Okay. Next slide, please. And I'm just going to give you an example of the very simplistic diagrams I used. And it's important to remember this was very conceptual. It wasn't based on much empirical data because there'd only been a few studies. I was trying to make it general and conceptual. So let's just look at model 2A, the phagotrophic algae that eat to obtain inorganic nutrients, versus 2C, the phagotrophic algae that are primarily phototrophic but eat when an irradiance is limiting. And I looked at responses of, well, I plotted responses I would expect of photosynthesis and feeding to dissolved inorganic nutrient availability, and also responses of photosynthesis to food concentration and of feeding to irradiance. And the big difference between the two groups is, one, in the algae that are primarily photosynthetic but respond to irradiance for feeding, is that food concentration shouldn't affect photosynthesis. And if we look at feeding in response to dissolved inorganic nutrients, it should decrease with nutrient availability in algae that feed to get inorganic nutrients, whereas in the ones that feed in response to light limitation, dissolved inorganic nutrients shouldn't have much effect. If we look at feeding in response to irradiance, in the ones that eat to get inorganic nutrients, you would expect that to actually increase with irradiance, because if they're photosynthetic and fixing carbon, then they need nitrogen and phosphorus to go along with that to actually produce more biomass. And so that should create a need for more nitrogen and phosphorus. So feeding would be a response. In contrast, the ones that feed due to limitations of in carbon uptake due to light, as irradiance increases, feeding should decrease. So this is a very simplistic way to look at this. I'm going to skip the next slide and go on to some of the strengths and limitations of this approach. Well, the strength of the conceptual models that I did was that it included flagellate ciliates in Rhizaria, and that it also included inorganic nutrients as a factor influencing mixotrophy, and it distinguished between light limitation and inorganic nutrient deficiency in driving mixotrophy. But there are some big limitations to this conceptual model. It's hard to actually apply in the real world because for most taxa, there's no or very, very limited data on feeding and photosynthetic responses to nutrients, irradiants, and prey. We still only actually have that data for very few species. And therefore, if you know something's a mixotroph in the oceans, you can sort of guess that it might fit in one of these categories, but you actually don't have usually the data to put it in one of the categories. So I think that's a big problem in actually assigning new species or species that haven't been studied very much to one of these mixotrophy types. The other thing that I've realized more and more since then is that mixotrophs can have mixed strategies that don't really fit these types neatly. One example of that I still am not sure what's going on is that, like many of the mixotrophic dinoflagellates that I worked on in Chesapeake Bay, feed primarily in response to nutrient limitation. And if you nutrient limit them, they start eating cryptophytes and other small phytoflagellates, and sometimes even ciliates. But if there's plenty of nutrients and light, some of these feed a little bit, but most of them don't really feed. When Hae-Jin Jung, who's worked a lot on dinoflagellates in Korea and mixotrophy, has said to me, well, these conceptual models are really wrong because I studied them in Korea in the wild, and they're always eating. And I think in most of his studies, he also looked at feeding on bacteria and Senecacoccus and small things. So why? What could be the explanation for this? Well, I'm not sure if, it might be a difference in technique and also in what you're looking at them feeding on, because maybe a lot of mixotrophs feed a little bit all the time on bacteria and Senecacoccus if they're around. Many of these mixotrophic dinoflagellates are difficult to grow in culture if you try to do it azenically. So maybe it's a mixed strategy between feeding to get trace factors all the time and then feeding to get nitrogen and phosphorus on bigger prey if you're nutrient stressed. So I don't know the answer to that, but that's like one possibility of a real mixed strategy in these dinoflagellates. The other thing that's come clear to me is that looking at rates may not be the best thing to describe these types. Perhaps changes in contribution of phototrophy and heterotrophy to metabolism would be better, or looking at growth like Harriet Jones did, because like recent work by Maria Massielli on Strombidium species that retain plastids, she found that if you starve them, often the photosynthetic rate didn't really go up that much, at least in some species, as you would expect from my conceptual model. Rather, it stayed about the same, sometimes went up a little bit, sometimes went down a little bit. But the one thing that always happened is that the percent contribution to their metabolism went up when they're deprived of prey. which would fit better than just looking at rates. Well, one thing that's sort of nice is that if you look at Harriet Jones's and my scheme for these classifications, they're both based on physiology, and actually they match up quite well. Her groups and my groups are really quite similar. There's really no real conflicts in there. Well, to sort of end, there are different ways mixotrophy has been used in different scientific fields, and even within oceanography, and also there are different classification systems, so it can be very confusing. And one suggestion I have is that when you use mixotrophy in a paper, you sort of explicitly state or reference how you're using it. So I think it's perfectly fine to limit mixotrophy to a combination of phagotrophy and phototrophy, but say it or reference a paper that says that so people know what you're doing. The other thing, I think it's very handy for readers if you cross-reference the mixotroph classifications. And in a review paper I did a few years ago, table 1, which I'll just put up. I'm not going to go through it, should help because
Diane, could you wrap up? Sorry, could you wrap up in the next minute or so? We're running a bit long here. Sorry.
Okay. So anyway, this table is there, and you can use it if you want. Okay? Thank you.
All right. Thank you very much, Diane, for that presentation. I don't think we'll have, unfortunately, time for questions, but maybe if people have questions, they can put them into the chat, and Diane might be able to see them. She's on her iPad. Are you able to see the chat?
I'm gonna somehow end my video, and then I think I can see the chat.
Okay. All right. Thank you so much again.
Sorry, I ran over.
I'll forgive you. Don't worry. So Kevin Flynn is up next. Nice enough to join us all the way from the UK today. So I guess it's this afternoon for him. Kevin, are you able to share your screen
Yeah,
at the
it's
moment?
Yep, okay. Apparently I've just been made the presenter, so let's see what happens. If you're going to share the right one is the question. Why does it always show me? Okay, let's see what happens. Okay. Okay, there's quite a lot here, so I will share as a PDF afterwards because I will sort of flick through this quickly. Can you see my screen at the moment?
Yes, we can.
Okay, so hopefully, if my computer's going to play ball. Okay, can you see it still?
Yes.
Yeah.
We're seeing a slide that says mixotrophic plankton progress.
Plymouth Marine Laboratory across the top. Okay, so very quickly. So I'm going to talk about mixotroph versus mixed plankton, perfect beast, lever human, basically how we got to where we are and why we made the decisions we did, which follows through the mixotin and then the mixed plankton paradigm. Okay, so we heard from a bit about this earlier. So osmotrophy, use of dissolved organics. Phototrophy, use of light, DIC, and typically DIN and DIP. And then phagotrophy, use of particulates, which of course usually involves killing other organisms, which is important from a trophic point of view. And then mixotrophy is then defined in different ways by different people, but generally it's a use of inorganics and organics. So it's in this context, it's phototrophy plus osmotrophy and or phagotrophy. So we either have this intersection here or this intersection here. So photoosmo mixotrophy, such as the use of dissolved free amino acids, this has been studied for decades, you know, from the 1960s or probably before. So I did my PhD on this sort of stuff in the 80s. So yeah, tons of stuff from back then. And interestingly, we can see parallels in what we found out about those sorts of things in sorts of things that Diane's been talking about. So, for example, if you stress a datum of nutrient or nitrogen nutrient or light or both, you get different responses. You get synergistic responses, interestingly, because bear in mind, amino acids are carbon and nitrogen, just like eating a prey item is getting a mixed meal, so is amino acid. So we see some sort of parallels in this. It can't be switched off by adding ammonia. And the control is the demand for amino acids. It's not for making amino acids like it is for ammonium nitrate. It's actually for the actual amino acids. And as I say, eating a food package is the same as eating a package of amino acids. So we can see some similarities here. We did a whole load of studies on this sort of subject, and basically we see that the use of amino acids looks like a leak recovery exercise. So it's not the same as eating, for sure. So osmotrophy is confused by excuse me, is confused by leakage and recovery of metabolites which you leak. As Diane mentioned, osmotrophy is ubiquitous in single-celled microbes, and photoosmomixotrophy is ubiquitous amongst phytoplankton. So mixotrophy really is confusing. Mixotroph versus mixoplankton then. So when we tried to explain what we actually meant by mixotrophy, as in eating, and phototrophy, we started confusing people. And we found this both with talking to scientists and also talking to the media and to students. You know, explaining concepts. This is a complicated subject. You know, it requires simplicity in terminology without too many caveats and without too many exceptions. And the use of mixotrophy are ambiguous. We need to differentiate between photoosmo versus photoosmophagotrophy in the context of this discussion. So we hunted around and we came across this term in fish base. We don't know who put it in there, but it is defined in fish base as planktonic organisms that can be classified at several trophic levels, which seem to tick the boxes quite nicely. So what we did was rather cheekily, we repurposed it and strengthened the definition. So now looking at the figure I showed you before, the Venn diagrams, phytoplankton are defined as plankton which are photo and osmotrophic, but they're incapable of phagotrophy. So they're in this section here. So that includes cyanobacteria, for example. Protozooplankton, they're capable of phagotrophy and osmotrophy, so that's in that section there. And then our mixoplankton are capable of photo, phago, and osmo, so they're in here. These sections are void, so there's nothing which is phototrophic and can't do one of the others, or is phagotrophic and can't do one of the others. But up in osmotrophy, we have bacteria, fungi, for example. So our phytoplankton cell looks like this with all these ins and outs. Our protozooplankton looks like this. And of course, our mixoplankton then puts the whole lot together. Lots of potential flows in and out. So photo, phago, and osmo in one cell. And then the balance of the different trophic modes are achieved by an internal physiological regulation. When we started sticking these things together, just to explain where Perfect Beast comes from, this is Don Henley of the Eagles, and he wrote this album, Building the Perfect Beast, which just happened to be playing at the time we were sticking these things together. So that's where that comes from. We developed a series of mechanistic phytoplankton models, variable stoichiometry, acclimative, temperature linked, telemetry linked, blah, blah, blah. Osmotrophic. DT Mitra had done similar sort of things with zooplankton models. And we basically stuck this lot together to make this Perfect Beast. So the Perfect Beast model is a complicated model. It's a mechanistic model merging facets of phytoplankton and protozooplankton models. It includes some processes actually which do not exist because we built it in ignorance of what we later found out and what others later found out. It also does not include some processes which do exist. It's been deployed in a series of experimental and ecosystem scenarios to some good effect, though. It had its first outing back in the GeoHab meeting in Galway. And back then we'd established, as Diane said, the way that mixoplankton works, it is not just adding these two processes together. These processes mix and match together between the elements and the physiologies. And we put various other products out using the perfect beast, in some cases tuning to data series, in other cases, yeah, pure theoretical biology, really. But all the ones on this page here have been fitted to either experimental or field data. And this last one here, the latest one by Leilis et al., this really shows how much you can do with these sorts of models, fitting across a wide range of very different organism groups using the one model structure. How does this fit in with the Jones-Stoecker response curves? Well, actually, we can describe any or all of those response curves using the one model. And they appear actually as a continuum. One construction can do the same by basically moving the way the physiology interacts. So you can make the physiology more or less sensitive to running out of carbon or nitrogen or phosphorus or anything else you want to. So any of these can be tweaked, but basically along a common set of feedback processes of the sorts which we know drive physiology. And of course, in nature, we need to remember that low nutrients are often allied to low light. So self-shading by other prime producers gives you food. Or you may be living low down in the water column, again, likely where you're surrounded by food as well. So many of these instances, trying to separate nutrient versus light stimulation of mixotrophy is artificial in a way, because many of these things are going to co-occur. Since 2009, well, of course, like many people, of course, we rushed off trying to get grants and failed miserably. To be told repeatedly the subject is not of significant, it's not significant, not sufficient importance. And although it's been studied over decades, arguably there is no coherent focus. Every new paper seemed to put new levels of complexity and novelty. And this did not help trying to squeeze in grant applications on a few pages. Biologists were looking for differences, modelers wanted to stick everything together. And eventually we specifically went down a line of going down a model focus. And the reason for that is basically only properly constructed models can really answer the fundamental question as to whether these processes are important or not, or where they are important. So we got funding from the Leverhulme Trust, and these are the grant holders for that. And over the following 4 years or so, we had a series of meetings. The first one being in Kalmar. And one thing you'll notice from all this is how we all gradually age over the time. So you can spot people and see what they look like. See that some people, like Diane, don't really seem to change too much. And others, like me, do. Some of these meetings had experiments associated with them. So this one produced an experimental piece of work as well. We had 2 meetings in Swansea. Again, pulling in other individuals. And from the Swansea meeting in particular, we put out this paper in Plankton Research, where basically we put forward the dichotomy which most people use, or the modelers tend to use in particular. By modelers, I include conceptual modelers, so that includes probably most of you. And of course, we drew it and said, well, no, it's not like that. It's like this. We then have a meeting in Horn Point with another collection of famous and dubious individuals. And from there, we generated more outputs. And this is where Susanna's work started to work with all the others to bring together information which basically showed these organisms are of some form or other are everywhere all the time. We then had a meeting in Swansea specifically for modelers and generated some papers. Some of us generated papers out of that as well. The outcomes from Leave a Human, basically we generated a common set of themes focused by the need to generate models, a series of multi-author papers on the importance of mixoplankton, and a common language, in particular a revised key, which I'll flick through in a minute. But the important thing about that key really, and that the whole effort, was we brought together what appeared like a myriad of different types of conflicting types, grouped around critical differences in their physiology. We identified specific gaps. Most basic, and this gap still exists now, is frankly the total inadequacy of most papers reporting activity of these organisms to actually provide data for modeling. There's very, very, very few papers which actually do a good job of this. This is the Protist paper, and I've updated because back then we were still using mixotrophic rather than mixoplankton. So this is the key. So we start off with protists and we ask, are they capable of carbon fixation? And if no, we rush off and label them as protozooplankton. If they are capable of carbon fixation, we then ask immediately, can they phagocytose? If they can't, then they're photoautotrophs. These are the phytoplankton, what we now label as phytoplankton. And only those organisms should be labeled as phytoplankton, as what came out from this. Yes, we now come into mixoplankton. We now ask, have they got an innate capability to perform carbon fixation? If the answer is yes, then these give us what we termed constitutive mixoplankton. So these have got all the genetic coding for actually making the chloroplasts, controlling them, photoacclimating, etc., etc. If the answer is no, then they acquire their phototrophy. Basically, this means stealing chloroplasts from their prey for different periods of time. Do they need specific prey? If they don't need specific prey, then these are generalists, non-constitutive. So they're non-constitutive in that they can't do it on their own. They're generalists not because they can steal their chloroplasts from anything, but that they don't have to steal it from just one type of prey. Otherwise, you have specialist non-constitutives, and we split these into plastidic or endosymbiotic forms. We've made a version of that to cater for the harmful algal bloom people, which came out actually this month. So in this one, we've labelled it up actually with different HAB species. So that might be of interest to some of you as well. Some points from this: mixoplankton are not all the same, and that we can't have a single, all those different types cannot be correctly modelled using a single fixed description. Neither can all mixoplankton within a type be modelled correctly using a single fixed description for the reasons that Diane has already mentioned. Whether these are functional types depends on your model setup and how you want to describe functional type. Because of the functional type can be split all the way down to a species level if that's what you wish to do, or is it photosynthetic at the other extreme. From Leverhulme, the Mixotin project developed. That's a 3 million 4-year European funded project, which is now in its final year. It's produced a range of training materials which will be made public over the next year. Training manuals like how to grow things, how to isolate things, how to model them, how to do genetic work on them, etc., etc. Like all such projects, of course, we start off with high hopes of answering questions. We end up with more questions than we started off with, but that's science for you. Aditi's now got a new funding line, which is funding Mixahub, which is a successor to Mixotin. So yeah, Mixotin, as I said, covers Europe. It's got its own web page, and that's being used for other things as well. So this is mixotroph.org. So various things are on that web link. The 2019 paper, this is the paper where we finally bit the bullet, having gone around in circles trying to justify mixotrophy, and came up with mixoplankton to solve the issue. And a whole lot of other papers have come out from Mixotin and will come out over the next couple of years. So the mixoplankton paradigm then, so over the last 5 years or so, 5-10 years, we see a long delayed acknowledgement of the importance of these organisms, the sorts of organisms which Diane and people have been working on for a long time. Yeah, we now bring it all together and see that collectively these things are indeed important. They're not insignificant. It's common throughout the protist phytoplankton, with the exception of diatoms. And when you start telling people, look, Emiliania huxleyi eats bacteria, even though it's covered in coccoliths, people will then actually start listening to you, that this may actually be important. It's not just oligotrophic, not just eutrophic, but everywhere. Size ranges, they cover the whole size range, from the smallest protists to the largest ones. So they're not confined to just one group allometrically either. From a food web sort of view, we go from this sort of level, when I call traditional here, of course, I've already included the microbial loop and the viable shunt, to this sort of level of complexity. And now we can see things labeled up in different ways, with different trophic implications. So 100 years of marine science, countless grants, research papers, climate change models projecting the future life on Earth, 2 series of BBC Blue Planet. All of them somehow managed to miss this. So there's a lot of inertia out there to overcoming this. There's a lot of vested interest in not including mixoplankton in what you do. Convincing people of this runs at various levels. So Aditi put this out in the conversation in 2016, and this was translated into Portuguese and Spanish, amongst other languages, and got a lot of hits all over the world. We did a Frontiers for Young Minds on this, and Aditi also did an article in Scientific American, which also got into the revolutions in science and was identified by them, not by us, as one of the discoveries that could change everything. So there we go. I shall stop there. Thank you.
Thank you very much.
Okay, how do I get back out of this? Stop sharing.
Stop sharing. That's right. You found it.
Okay, I've got it. There we go. Sorry, that was quick.
There we go. Sorry, I had trouble unmuting. Thank you very much, Kevin, for that talk. Great. I'm glad to hear that Mixotin's moving forward in some form as well, and that you guys are going to keep working on more stuff to come, and looking forward to reading all of the papers coming out of the Mixotin group as well. So we've unfortunately probably don't have any time for questions, but luckily we should get going into our breakout groups. And so you'll be, you guys are already randomly assigned to groups, and you'll have myself, Holly, Jessica, Karen, or Becky will be one of the people in there. And there's been a set of questions that Diane submitted and Kevin approved to start a discussion on this topic amongst everyone. And then we will come back together in about 25 minutes or so and have a wrap up from there. So I'm looking forward to seeing what everyone's discussion looks like, and see everyone back here in a little bit. Oh, someone's editing the breakout rooms right now. Oh no.
So just while we're waiting to get sorted, the idea here is that we'll talk in smaller groups about the questions that Diane very kindly listed for us. and come back at the end of the meeting to debrief all together in a bigger group. So as you're in your breakout room, take a glance at all the questions you could be discussing. We won't have time to get to them all in the depth that we'd like to, and cover what you can.
One place to another, rematerialize.
All right, welcome back, everyone. I hope everyone had a good discussion. It had to be dragged out at the last second from the breakout room myself. So if we want to start with a very, very short report from what each of the different groups discussed. I'll start with Holly. I think you were, I don't know what
Sure.
group you were, but yeah, go.
Yeah. So we had a really stimulating discussion that quickly diverged from the question prompts and turned into essentially the importance of wrestling with carbon, even if it might not be the only axis on which we differentiate mixotrophs, because of the fact that we had some really skilled empiricists with us who were talking about some of the methods that we could actually use to trace carbon's flow through cellular physiology and better understand how mixotrophs are actually doing what they're doing. And that turned into a broader discussion about whether important ways to differentiate mixotrophs might all boil down to better understanding some of the trade-offs involved in a mixotrophic lifestyle. And if we could accurately estimate the different costs that mixotrophs experience, such as the cost of constructing metabolic machinery, of maintaining it, of utilizing different things that you take up if you're a mixotroph, and also understand the environmental conditions that the mixotrophs are in, we might be able to come up with sort of a unifying framework for mixotrophy. But of course, this is complicated by what we all understand about not only the nuances across different mixotrophic taxa, but also within a mixotroph's lifetime. Different parts of the life cycle, whether a mixotroph is blooming or not, might also affect what it looks like from the perspective of its carbon and nutrient budget.
Very, very interesting. That's great to hear that the conversation went off in such a direction. If we want to move on to Karen.
All right. So we said it should include everything. We said no. We should include everything. And we had a little bit of a discussion that it's really semantics and that it doesn't matter, but someone should make a decision and stick with it. And there was also a thought that we could have a very general definition that everyone can just stick with and agree with, so there's less confusion in the world. But then as we learn more, we could have subcategories under that with very narrow definitions that are very clear. So yeah, that was kind of where we landed.
Did you guys discuss anything else?
Oh, sorry. Yeah, we did. So then, are we going through each? You want me to talk about all the questions now?
Just, yeah, real quick summary of
Sure.
everything.
So for the functional types, I think generally we like them. There was some concern that they might blur a lot of diversity, that the Jones-Stoecker definitions maybe help include. But we'd really decided it really depends on your question or the purpose of these functional types, and that they could really be they could be ecologically emergent as you're studying these things. And we also learned from Kevin that there's a really, really important ecological difference between these generalist non-constitutives and specialist non-constitutives. So even though they seem similar, they're very different. And we didn't really get to the third and fourth questions per se.
That's okay. Jessica, you mentioned that you guys managed to get to every question, actually. You might have been the only group that did that.
Yeah.
You want to touch on?
Yes, so our group, I guess breakout room 3, did get through every single question, but mostly because I think the first couple questions, we said that we should stick with carbon as a currency. It really determines how we define autotrophy versus bacteria and other osmotrophs. And and we use it in models. And our group actually, as we went through, really struggled with the simplicity versus complexity definitions, the splitters versus the groupers in biology. And our group tended to err more on the groupers side of things. So we didn't think that we needed to add more functional types in question 2 than the ones already within Mitra et al. 2016. We thought that the distinctions from Jones and Stoecker were important, but functionally, how are we able to distinguish between what different types of mixotropes are actually doing in nature is going to be very complex. And there's also some species-specific interactions that are important, but we know very little about what individual organisms are actually doing. And so it would be difficult to maintain these distinctions. And then we spent the most of our time on question number 4. And there was an interesting point that was made, from the prey perspective, what makes them susceptible, what makes them able to be exploited or susceptible to capture, and then how can different species, on the flip side, the predator side, how can different species exist or survive if there's such specific food requirements? We talked about, within the context of trying to understand global change, effects of global change on marine ecosystems, whether these specificity or generality of predator-prey interactions, understanding that would be important, kind of in this shifting phenology question. And that there was an interesting distinction between kind of pelagic ecology, which comes from this global biogeochemistry perspective, and benthic ecology, which is much more community-based. And this mixotrophy work is really showing the importance of community ecology within the pelagic realm. That's it for us.
Becky, then?
Okay. Yeah. And it's really interesting to hear that so much of this group is in agreement on on our perspective. So yes, we agreed that carbon was the currency, but also that it's important to consider some of the other aspects involved in cell growth, and particularly as active processes in the definitions of mixotrophy. And also that we recognize the difficulty of lumping constitutive mixotropes because there is a spectrum within this group as well, but we're not sure how to describe these differences at this point. But it's important to recognize that even though they are a group, they do represent some level of variation. In terms of the Jones and Stoecker distinguishing between them, yes, we think they're important. And again, it relates back to that spectrum, but we're not quite sure how to deal with it yet. And finally, on the non-constitutive mixotropes, again, these are interesting. They're potentially very important, but it's going to depend upon what level you're going to look at. And everybody has a different perspective. So some of the things that we talked about as non-constitutive mixotropes is that and utilize their prey, and to figure out how much they do matter or which ones matter in the environment that you're studying. And this led to a short discussion of what Ben called parameter space. So we need to explore that a bit more in terms of, we've got information on organisms that are everybody's favorite that works well in the lab. We need to broaden that out and explore a little bit more to things that are more relevant in different environments and get a better handle on how these differences occur. So.
Great. Yeah, and I'll just wrap up really quick. Our group only got to questions 1 and 3. With 1, we were kind of in agreement with everyone else that carbon probably should be the currency that's used. It was brought up that there's a lot more complications when you introduce nitrogen, just the way that nitrogen cycles and is interacted around in the cell. That could really make that very difficult compared to carbon, which is a bit more straightforward. Uh, and um, and the fact that just for modelers to be as simple as possible for them, make things as easy as possible for them, and don't start introducing multiple nutrients and other factors to consider. And then for the third question, the Jones and Stoecker distinctions within the constitutive mixotrophs, we were also in favor of considering them. We talked about how there's a lot of body of research looking at those distinctions within mixotrophs out there. At least like a lot of species specific, looking at how they respond, which species respond to nutrient limitation versus light limitation, and that isn't just worth pushing aside. But of course, also looking at in the environment and on a community level, trying to figure out how to better understand that so that it can be incorporated into models, considering that these seem to be a driving force in the activity of these constitutive mixotrophs, these limitations, but trying to understand how to model that. So that brings us to 12:30. I hope for everyone else the time just flew. I can't believe it was 90 minutes already. Is there any last comments that people want to make here in the larger group on this topic?
I just want to echo Holly's comment that if people want to go into the Google Doc and add any thoughts that they have after the fact or correct anything that the note takers messed up, please do. And thank you so much for your participation. It was really fun.
Yes. Yeah. And thank you again to Diane and Kevin for taking your time to put those presentations together and come up with those discussion questions for us. They were very stimulating discussions, and we really, really appreciate that. And we will, just looking ahead here, June is the OCB meeting, and we'll hopefully having some event associated with that. We have a tentative date, but nothing confirmed yet. But then looking ahead, the rest of our meetings, virtual meetings will look very much like this one did today, albeit on different topics. And so the next topic will be biogeochemistry and mixotrophs observations, followed by in September, we'll be looking at biogeochemistry and mixotrophs and modeling. So jumping into a very different topic, but hoping for also very stimulating discussions. So thank you again, everyone. Hope everyone's spring is getting better as we move into at least a more vaccinated United States and trying to get back to normal, and everyone's feeling a little bit more at ease. We'll be in touch with updates as they come. Thanks everyone for joining.
Hey, thank you. It was really
Bye.
stimulating, fun.
Thanks, everybody.
Thank you, everyone.
Thank you.